The overall goal of this procedure is to generate linear, high aspect ratio micro and nanocomposites using copper-containing starting materials and cystine. This is accomplished by first combining sonicated copper nanoparticles or copper sulfate with cystine and water in a sterile vented culture flask. The second step of the synthesis is to place the combined components in the flask in a 5% CO2 incubator at 37 degrees Celsius for at least 6 hours. Next, the flask is inspected by eye and by white light microscopy to determine the extent of linear structure formation. The final step is to terminate the synthesis through placing the synthesis flask in a refrigerator maintained at 4 degrees Celsius. Ultimately, digital microscopy is used to characterize the synthesized structures.
The main advantage of this technique over existing methods like electrodeposition is that the described synthesis can be easily scaled up in liquid form, and the process occurs at physiological conditions.
The implications of this technique extend toward therapy or diagnosis of diseases such as cancer, because synthesized structures can be degraded by cells, potentially providing a means to deliver drugs.
I first had the idea for this method when we were carrying out experiments investigating the potential toxicity of different nanomaterials.
Visual demonstration of this method is critical, as the final synthesis steps can be difficult to learn. Incorrect procedures may result in aggregated structures or no structures at all.
To perform the self-assembly synthesis using copper nanoparticles, or CNPs,